Let's discuss how cancer results from mutations. As we've already learned, cancer is a group of diseases that share the trait of uncontrolled cell growth. There are more than 100 types of human cancers, and other non-human organisms develop cancer as well. Uncontrolled growth results in tumors, such as this 32-pound tumor that was removed from a patient in New York City by the surgeon shown here, Dr. Julia Teixeira. When we look at cancer cells, they have a number of abilities that normal, well-regulated cells do not have, which explains their growth into a tumor and eventually resulting in possibly death. Cancer cells, over time and via a series of mutations, develop these abilities that allow them to continue to grow out of control. This figure shows 6 major hallmarks of cancer cells. It's adapted from a famous paper published in the year 2000 by Douglas Hanahan and Robert Weinberg in the journal Cell. And this is a kind of way of thinking about cancer in a simplified way, which makes sense because cancer is many diseases, but they all share certain features. And these features are a result of mutations. Okay, so first, cancer can grow and divide without receiving a signal from the outside. So for example, do you remember the growth factor hormone and receptor? Well, what if we acquire a mutation that just turns on the signal transduction pathway all the time, even if growth factor isn't present? Now we're going to have a cell that's acting like it's getting the signal to divide all the time, even when the signal isn't there. Cancer cells also lose the ability to respond to external signals telling them to stop dividing. One possible way that could happen is if a mutation removed the functional receptor for an outside growth suppressor. Now the suppressor molecule might be there, but the cell has lost the ability to respond to it. Cancer cells also lose the ability to undergo apoptosis, and this is a major problem because now we have cells that continue to live and divide with damaged DNA, which means that the cell is likely now to accumulate additional mutations since it's no longer able to undergo programmed cell death. This is going to undermine the genetic stability of the cell. Cancer cells can become immortal. This is in contrast to normal cells, which can only divide a certain number of times. And that's mainly because the ends of chromosomes, called the telomeres, get shorter with every cell division. Cancer cells, in contrast, acquire the ability to lengthen their telomeres again, and therefore they're able to divide indefinitely. Angiogenesis is another feature that cancer cells acquire, and this is the ability to recruit a blood supply. Tumors are unable to grow very large, about 1 millimeter, without the food molecules and oxygen that a blood supply delivers. But once a tumor acquires the ability to secrete proteins into the environment that cause blood vessel formation, then it has the ability to grow very large indeed. Remember that 32-pound tumor? Finally, tumor cells acquire the ability to invade neighboring tissues and blood vessels, allowing them to metastasize, or in other words, form new secondary tumors. Metastasis is common in later stages of cancer and generally means that the possibility of treatment and survival are significantly decreased. Cancer is the result of mutations, and these are generally mostly going to be somatic mutations. Now, we can also have inherited mutations or germline mutations that are passed down from parent to child. We can think of these inherited mutations as predisposing someone to cancer. So maybe you've heard about genes like BRCA1 and BRCA2, which is how we abbreviate BRCA1 and 2, that can confer a high risk of breast and ovarian cancers in people who inherit certain mutations in these genes. Or APC, which confers a predisposition to colon cancer. Nearly all people who inherit certain mutations in this gene develop colon cancer by the age of 40. Another example would be Li-Fraumeni syndrome, and this is found in people who have inherited mutations in p53, which we sometimes call the guardian of the genome for its role in the cell cycle. People with these inherited mutations in p53 are susceptible to a number of cancers, often at a very young age, such as in childhood or early adulthood. Now, not all of them actually develop cancer this early, because p53 is a tumor suppressor gene. people with one mutation in p53 inherited from one parent must also acquire a mutation in their second copy of p53 before p53 is rendered unable to regulate the G1 checkpoint. Finally, mutations in RB1 can lead to retinoblastomas, which are tumors in the eye that usually occur in children. But cancer requires more than one mutation, so even people that inherit a mutation that increases their risk also need to acquire more mutations before cancer can occur, and these will be somatic mutations. And mutations in just any gene aren't necessarily going to do it. Cancer cells acquire mutations in genes that function in specific metabolic pathways that do things like regulate the cell cycle, including the cell cycle checkpoints, respond to growth signals, regulate cell death, etc. All right, let's put this into context with what we've already learned about cell cycle regulation. Recall that we have molecules that are able to positively regulate the cell cycle and molecules that are able to negatively regulate the cell cycle. And we need both to keep this process functioning normally. Here's our car analogy again. And here's a figure showing this more as a pathway. We have genes shown at the top, which code for proteins that regulate the cell cycle. Negative regulators of the cell cycle are called tumor suppressor genes, and positive regulators of the cell cycle are called proto-oncogenes. And if they're all functioning correctly, then so is our cell cycle. But what if we mutate the genes that code for the proteins that regulate the cell cycle? For tumor suppressor genes, we generally need to have both copies of the gene acquire mutations. Why? Because a single copy of a normal tumor suppressor gene is often enough to keep the cell cycle in check. As I mentioned, p53 is a tumor suppressor gene. And if we lose one copy of the p53 gene, we're generally okay. The remaining copy of the gene makes enough p53 protein to keep the cell cycle regulated, arresting the cycle at G1 if there's DNA damage and activating DNA repair proteins. And if the damage can't be repaired, activating apoptosis. But if we lose both normal copies, well, now the brakes are off, and the cell will keep dividing even though we have damaged DNA. And that is really bad news because this allows the cell to accumulate even more mutations. We call a mutation like this that results in a complete loss or a reduction in protein activity a loss of function mutation. On the other hand, a proto-oncogene generally mutating one copy is enough because these are positive regulators that stimulate cell division. And if we have a positive regulator that now has more activity, this will tend to increase cell division, so we don't have to mutate both copies. And it's like now we have the accelerator pedal stuck down and we can't stop it. Once a proto-oncogene has mutated to cause cancer, we call it simply an oncogene. A mutation that leads to an oncogene is a gain-of-function mutation. Gain-of-function is when a protein has now more activity than the original form, or it's taken on a new or novel function. Once we've accumulated multiple mutations, and this can be a combination of mutations in both tumor suppressor genes and proto-oncogenes, then it's possible we've progressed to a cancerous tumor.